Welcome to my Punnett Square review for Spotlight. So the first thing I want to introduce you to is to my little template that I've created for Punnett Squares. And I did this on purpose. So Punnett Squares, usually when you're looking at Punnett Squares, you're just looking at the square, right? But I've seen students struggle trying to figure out how to deal with this Punnett square and how to set it up, because once you figure out how to set it up, it's easy, right? It's the setup that's actually the problem. And so what I The way I set this up is by having, so this is the mother right there, and this is the father, and mother and father are diploid organisms. So mom is a diploid organism and dad is a diploid organism. So they have two copies. Diploid means they have two copies of every piece of genetic material. Now, this diploid organism, in order to create babies, they have to make a haploid um, egg and a sperm cell. So this down here, that's the haploid part. And in my and so this both of these, right? Both of these are the haploid parts. So this one here is haploid, and this one here, they're both haploid. And mama and daddy are both diploid. So we're going to keep this in mind as I'm trying to make another arrow to create for up there without this interfering with my little word over here. So this is my other little diploid little character. All right. So mom's a diploid. This process here to get from diploid to haploid, this process, this is a time arrow now, that process is called meiosis. Just in case you're trying to get connections between mitosis, meiosis, and all that kind of stuff. Phenotype is what somebody looks like. So if we're saying our mother has blue eyes, for example, and our father has brown eyes, that would be a phenotype. Phenotype is, this is what somebody looks like. A genotype is a letter combination. If you're talking about a single trait, a genotype, it's always two letters. So it's always going to be something, I'm going to put a text here. So something, um, a letter and a letter. And father is also a letter and a letter. So it's always two letters, whatever those letters are. Now, those letters, there are conventions that are associated with those letters. So those letters, there's a convention that if that letter, maybe I should have used um, L for letter, if that letter is if it's a capital letter, then that's the dominant gene. And if it's a lowercase letter, no, seriously, a lowercase, no, I mean it with the lowercase letter. You don't need to correct this for me. Thank you so much. Then that's the recessive gene. Dominant and recessive were coined by Mendel. And dominant recessive doesn't mean anything about better or worse or anything like that. It simply means that if you have the dominant gene is the one, um, or the dominant allele. I guess I should really call it allele, not gene. Dominant allele and recessive allele. So the dominant allele is the one where you just need one. Like, so you have two genes each time, right? And you only need one of the dominant allele in order to be able to have the dominant trait expressed. So in order to ever see the recessive trait, you have to have two little n's, right? Two lowercase letters. And so we're not going to practice with some of this stuff. Now, this slide set is available to you. Now, obviously, because I have a video, it's hard for me to pass this along to you. But I'm going to try to link it. But if not, if all else fails, you can type in this letter combination and you get to an editable copy of this slide set. But once you get to that slide set, either by me having given you a copy or by you haven't acquired an editable, you've figured out how to type this thing in, then what you can do is you can play with these things on here. And so the story I always tell in class is the mother phenotype has brown eyes, and the father phenotype has blue eyes. And eye color, so eye color, I always have to say that as a biology instructor, eye color is more complicated than that. But for our biology purposes, we just claim that big B, the brown, is dominant over little b. Now that's because big B expresses melanin, and blue eyes basically is when eyes don't have a color. It reflects the colors that, if there's no color there, then other minor colors are reflected in some way, shape, or form. And really, all you need is one brown gene in order to reflect enough melanin, to produce enough melanin in order for the eyes to be brown. Like I said, it's really more complicated than that, but brown is the dominant gene. So if we've got a father with blue eyes, I told you that in order for the recessive to show, you always have to have two of the recessive gene. So anytime you're trying to figure out anybody's genetics or whatever, start with, or you're trying to figure out a Punnett square, you're trying to figure out offspring from parents that are, where you only know the phenotypes, always start with the recessive parent. Because the father, he's got phenotype is blue eyes, so he's got to have two of these Bs. Now, the way you're going to make more of these, you're just going to highlight them. You're going to go Control C, Control V. And with Control C, Control V, that'll copy them. And so you've got dad. So this is your diploid dad. And diploid dad now makes sperms. And his sperms, because all of his genes that he's got available to him, all of the alleles that are available to him are little b's. All of his sperms are going to carry little b's. Now, mom has brown eyes. Because she has brown eyes, we know for a fact she's got to have at least one big B, right? Because the big B is what makes her have brown eyes. We don't necessarily know what her other gene looks like. We know she has two of them. So she's either going to be big B, or big B, little b. In order to figure out what she is, you would have to ask her what her for what her parents' phenotypes are, what her parents' eye color looked like. Now, if she tells you both her parents have brown eyes, you have no more information than you had before. But if she tells you one of her parents had blue eyes, then you suddenly have more information, because if one of her parents had blue eyes, then you have that same situation as here, where that parent could only pass on the little b. And so now, for just entertainment purposes, let's just say she had a blue-eyed parent, so she's big B, little b. Um, in her egg cells, which is the haploid, right? Her cells undergo meiosis at some point, and she produces egg cells. These egg cells can carry either a little b or a big B. So now you've got the setup on the outside of the Punnett square. So now you know, so there's only, for one trait, there's always only one letter coming in, not two of them, because you've got the haploid trait that you're coming in there with. And so for her to fill out the Punnett square, all you're doing is you're dragging these things down. So Control C, Control V, Control C, Control V, Control V. And you're just going to drag them across to fill the square. And then the same here. Control C, Control V, and there you go. So now you're looking at the Punnett square, and the Punnett square now tells you the genotypes, right? So the genotypes are BB, big B, little b, and little b, little b. And now you interpret, based on what you were told about dominance and recessiveness, you interpret what is the outcome now, right? So you interpret that, okay, so these two here are big B, little b, so they're both brown eyes. And these two here are blue eyes. They may be boys, girls, whatever, right? There's nothing that says about that is said about this. We just know it's about 50% brown eyes, 50% blue eyes. Now pay attention to the questions that we're asking you. If I ask you about a phenotype, answer with a phenotype. If I ask about a genotype, answer with a genotype. If I ask about percentages, make sure you tell me something about percentages. Sometimes giving me more information, if you start giving me wrong information, will lead to points being taken off. So you can practice this to your heart's content with any kind of letter combination that you desire. One of the ones I want to practice with you real quick, just in terms of the interpretation of these. So the letters D traditionally, and I'm going to just, because I just really want to use it for practice. Traditionally, big D and little d are being used for two different traits. One is dwarfism, and the other one is deafness. And it's just traditionally that's the letter combinations that are being used. And so I'm just going to fill this thing in for two heterozygous parents. So they're heterozygous for big D and little d, and I'm going to leave it um, later on. We're going to tell you what big D and what little d is. So I'm just going to fill this in just so that we've got this Punnett square filled in just exactly the same way we had this before, right? So we're just dragging mom down here, the little d from her egg and the big D from her egg. And then your odds of having the children is going to be, so if you have two heterozygous parents, each parent in their egg or sperm cells has a big D and a little d, and so the odds for the children are that you've got big D, big D, big D, little d, big D, little d, and little d, little d. So those are the possibilities. Now, this is always the same. Punnett squares are set up, it's very mechanistic. It's always set up the same way. Where the actual thinking happens is when we ask you about phenotypes. So let's say, for just sake of argument, what we're talking about here is we're talking about deafness. Deafness is a recessive disorder. So In this case, mom and dad are heterozygous. Little d, because deafness is a recessive disorder. The deafness gene, the deafness allele, by definition, is the little d. So you have to have two little d's in order for you to be deaf. Now, in this case, you have a mom that has a little d. She's hearing, right? Her phenotype is she's hearing. You've got a dad. His phenotype also is he's hearing. But because both of them carry a little d, they're carriers of the little d. Some of their sperm and some of their eggs are going to have little d. And so there is a possibility that a quarter of their children are going to be deaf because they are homozygous for the deafness gene, little d, little d. This one here, big D, he's hearing. She is hearing. Has no chance at carrying on the deafness gene. These two here are heterozygous just like the parents. So those are hearing. So 3/4 of the children phenotypically are hearing, and 1/4 of the children phenotypically are deaf. Now, let's talk about same setup. We're going to talk about dwarfism. And dwarfism is an interesting one. Dwarfism is a dominant for dwarfism, but it's a recessive lethal. So it is lethal in its recessive state. Recessive lethal, little d. So if you have just one big D, not little, yes, never mind, scrap that. So dwarfism itself is dominant. Achondroplasia is dominant. So if you have one big D, then you actually have dwarfism. And actually, we can't say little d, little d. It's recessive lethal. Recessive lethal like that. Yeah, that was the wrong explanation right here. So dwarfism is a dominant, but it's a recessive lethal. So in order for it to be lethal, you have to have two big Ds. Only in that case is it lethal. So it's not lethal if you just have one or the other. So same setup. So this person here, because dwarfism is dominant, this person is a dwarf. This person has short stature. This person also has short stature. Both of them carry on their dwarfism genes because while it's the gene for dwarfism, it is a dominant gene. So all you need is one of them to make it. So it is dominant, even though it's not better, stronger, anything like that. It is dominant. Dominant is something. It's just a physiological. It's not a judgment in any way, shape, or form. It's just a physiological state. So the dwarfism gene um, is carried along in some of the egg, in 50% of the eggs and the sperm cells, and they distribute exactly the same way as the deafness gene, and then comes the interpretation. So the interpretation then is two dwarf parents has 3/4. Well, actually, let's start with the other ones. 1/4 odds of having an average height child, 50% odds of having a dwarf, and 1/4 odds of having a embryonic lethal child. So that is not survivable. Little d, big D um, is an embryonic lethal. So in this case, the child will not survive. And parents usually test for this. So if you are an achondroplagic dwarf and you are having a child with another achondroplagic dwarf, then you need to have genetic testing done on your, or it's it's a good idea to have genetic testing done on the embryo. If there are different types of dwarfism in mom and dad, that may not in fact happen. So this is, there are the idiosyncrasies and the tricks usually happen right here in the in the interpretation of the phenotype, more so than the setup of the Punnett square. Because as you saw, I didn't have to move any letters between the deafness situation and the dwarfism situation, and the interpretation was completely different because the Punnett square is very mechanical. It's very mechanistical. Now, this mechanics sometimes involves more than two alleles. So both of the stories before involved two alleles, a big B and a little b, a big D and a little d. Sometimes you have more than two options, because these options, as you remember, they are, if you have a chromosome like so, um, and a diploid organism starts out with two chromosomes, then all of the A's, O's, big B's, whatever they are, those are going to be at a locus, right? So there's going to be a locus on this chromosome, and this locus is going to be at the same place of each chromosome. Now, whatever is on this locus, the value that this locus can take on in this particular instance is A, B, or O. So there's three options for the locus, but you still have only two chromosomes. So your genotype is still only going to be two letters. Okay? But now you have three choices between each of the two letters. So again, now per convention, I don't even have to tell you anything about what this is, ABO. It could be anything. Per convention, you already should know that A and B, because they are capitalized here, those are the dominant ones, and O is the recessive one. Mother has the phenotype of O, so that's the recessive one. So it's clear, because she has a genotype of O, she's got to have two of the little o's, even if you have no idea what this is. Now, the father, his phenotype is AB, and again, without knowing what any of this entails, you can say, all right, father, AB, all right, wonderful. I have an A, I have a B. He probably has to express both of it. And then we also know that his sperm, some of his sperms are going to have the A, some of his sperms are going to have the B, and some of it, and mom, oh, this is going to be this way. All right. Now I can tell you what this is. So what this is blood type. Blood type, we have three different alleles that we contend with, A, B, and O. The expression of these alleles, what blood type basically is little flags on the outside of red blood cells. And in the case of the A blood type, the gene for A basically plants an A flag on the outside of the red blood cell. And the B plants a B cell on the outside of the red blood cell. And the O gene plants no blood type, no flag on the outside of the red blood cell. This is why O is recessive, because O is the absence of a flag. So if you have the A flag, you've got the A flag. If you've got the B flag, you've got the B flag. If your genetic setup is A and O, you're going to present the A flag and no flag. So it's going to look like phenotypically like you've just got the A flag. Okay. So let's drag this along and just see what kind of, the genotypes are easy, right? The phenotypes are going to be harder. So, well, not really harder, but that those are going to require interpretation. So, and mama. So a mother that has a blood type O and a father that has a blood type AB, they are going to have children that are all going to have what blood types? So you have AO here, AO, BO, and BO. So those are the parents, the children's genotypes, possible genotypes. So this here, AO, they'll have the A flag and the no flag. So that'll look like they'll just have the A flag. And so those are blood type A. And these guys down here, those have blood type B. Which is kind of an interesting thing, right? Because in this particular case, the parents have completely different phenotypes from their children. But that's how genetic works. Now you can play this any way you want to. So there's another set here for you to play with blood types, however you want to. But the take home is the setup is always the same. You start with the diploid cell, you separate it out into the two haploid parts, because that's what happens in the cell. In the cell, you end up with a diploid cell, meiosis happens, makes two haploids, and then those haploids are independently assorted with the other haploids that the other parent made. There's one more special case I want to go through with you guys, which is the sex-linked inheritance. So sex-linked inheritance is inheritance that is linked on the X chromosome. In 99.9% of the cases we're going to ask you about, anytime anybody says sex-linked, it's X-linked, because the X chromosome is so much bigger than the Y chromosome. There are hardly any genes on the Y chromosome at all. There are tons of genes on the X chromosome. Now, hopefully we know that women are XX. There are variations possible, but that's the standard. The standard is women are XX, and so they're passing on in their genetic material the X's. And men are XY, so they're passing on in their genetic material an X and a Y. And then without knowing anything else, we know that the mother passes on her X's, X number 1 and X number 2, to all of their children. And so she does, as we all know, not determine the sex of the children, right? The father, he passes on the X to half of his children and the Y to the other half of his children. And so the thing about sex-linked inheritance is you got to watch out that the trait goes along with the sex of the offspring. So, and the parent too. So in this particular case, and again, this was very mechanistic, right? I started out with a diploid mother that has XX. I have a diploid father who has XY, and then we're just passing down these genes. If I tell you X and Y, that's not a problem. Most of you get this right. It starts getting interesting when we give you tasks where we say, hey, let's go talk about hemophilia, which is X-linked. Or let's talk about hemophilia, or red-green color blindness. So let's talk about those two. And then all of a sudden, I get crosses. Both of those are sex linked. And I suddenly get crosses from students who are like, well, there's big H and little h. And I'm like, no, don't use, if it's sex linked, you're going to have to use X's and Y's. So let's say, for example, we have a mother whose phenotype is that she is, so we're going to talk about, hang on, just make sure, we're going to take green color blindness off the thing. We're just going to talk about hemophilia here for a moment. So we're going to say mother is XX, and she is not a hemophiliac. So her phenotype is not hemophiliac and not a carrier. And let's say the father is a hemophiliac. So since the X is so the mother is not a hemophiliac and not a carrier, so she's got two perfectly fine, pristine X chromosomes. The father is a hemophiliac, we said. So the father has a broken X chromosome. So his X chromosome, this one right here, that was perfectly fine, we're going to have to take that away, right? The perfectly fine X chromosome, and now we're going to take this perfectly fine X chromosome away because he's a hemophiliac, so he's got the broken X chromosome. So here's the broken X chromosome. Here's the broken X chromosome in the sperm. And then here's the broken X chromosome as it's being passed along to each of the individual matings. And as you can see, his sons, the father's sons, are not affected in any way, shape, or form. The father's daughters are affected, but none of them are hemophiliacs. What they are instead is they're carriers. Carriers means you carry the broken gene, but because women have, females have two X's available to them, and only one of them is inactivated. They're inactivated by random in each body cell. And since each of them is inactivated randomly, they're not affected by hemophilia. They're just carriers. So I'm going to undo this here real quick. So this is dad. Dad passes this on to his daughters. I'm going to put this back here for a second. Now, what if mother, let's say one of the daughters, one of the carrier daughters passes, has children with a male that is perfectly fine, does not have hemophilia. And I guess we should delete these here so that nobody gets confused about what we're looking at here. So this time we have the mother that's a carrier who has children with a male who is perfectly fine. And so these are two parents who can, their blood clotting is perfectly fine. There's absolutely nothing wrong with them. There's nothing, they had no idea. And then one of the all the girls are fine. They look fine. Their phenotype is fine. One of them is in fact fine. One of them is a carrier. One of the boys, so a quarter of the boys, also looks fine and is fine. But one of the boys here, this one XY over there, the one with the broken X and the Y, he does not have a anything to fix his X chromosome, right? He doesn't have a way to fix the broken gene, so he now has hemophilia. Now, for women to have hemophilia and or color blindness is much harder because you need both X's that need to be broken, which means you need to get the gene from both your mother and your father. So in this particular case, if the father, gonna kill his little X over here, if the father, for example, had hemophilia, so his little X's would be dragged along here too. And then you're going to look at this interpretation. So now you have a girl who's a hemophiliac, a female who's a hemophiliac, a female who's a carrier, a male who's a hemophiliac, and a male that is perfectly fine. So those are your options. So hopefully this is helpful. Hopefully you can, again, the take home basically is the setup for Punnett squares is very mechanistic. Set it up like this if you have if you're struggling figuring out what you have to put there. So start with the diploid organism. Think about the fact that what you're talking about is you're talking about these two chromosomes that are basically going, right? There's a chromosome here and a chromosome goes here, right? So these are the chromosomes that are splitting up from the diploid organism to the haploid organism. So each allele goes into one direction, and then they get reassorted when they find their partners. When the sperm and the egg cell find each other. So hopefully this helps, and let me know if you have any questions.